Okay, well, thank you very much. Thanks for inviting me to speak at this meeting. So I'm going to give a presentation, and I'm going to talk about two small sort of research projects that we've been undertaking in the last few years, looking at natural resistance to two different invasive Phytophthora species and management implications from forestry perspective. I would just quickly like to acknowledge the people involved in this work before I start. One is Heather Dunn, who's a PhD student, now finished, who did a lot of work with the larch. She was supervised by John Mackay at the University of Oxford. And then my colleagues at Forest Research, Carolyn Riddle, Dave Clark and Tony Clark. So just a few Phytophthora facts for those of you who might not be aware. Phytophthora is Greek for plant destroyer. They're not true fungi, they are oomycetes. They're water molds and they spread via free-swimming zoospores. And the top right photograph shows a spore sac of a Phytophthora releasing zoospores into water. So they're born in water and soil, and Phytophthoras can persist in soil for quite long periods. And this is a thick-walled resting spore, this middle photograph of a Phytophthora. Generally speaking, Phytophthora infect plants through the roots, but they can also infect through the stems and the foliage. There's more than 180 Phytophthora species described worldwide on a very broad range of hosts. And we have around 50 species currently known to occur in the UK. But the reason why we're so particularly interested in Phytophthora is the emergence of 6 new species from we don't know where, which are pathogenic to trees in the last 15 years in the UK. So I will talk about two quite contrasting host pathogen systems in some ways. The first one I'll speak about is Phytophthora ramorum, which you probably will have heard of because it also is having a big impact in Western USA on live oak. But in the UK, it's killing Japanese larch, which is an exotic plantation species and probably of quite low genetic diversity in terms of the host. And I will also then speak about another invasive Phytophthora, which is Phytophthora ostracedri, which is a root-infecting Phytophthora, which is killing juniper, which is a native species of considerable ecological importance in the UK, but also a species that is of high genetic diversity. So I think this also has some implications going forward when you're looking at natural resistance. So then I'll start talking about ramorum on larch. Phytophthora ramorum is an aerially transmitted Phytophthora. It's quite damaging on larch. Larch is deciduous, and as the needles start senescing in the autumn, the pathogen will infect the needles. You can see the top left-hand photograph here is showing clusters of dead and dying needles. These are infected with Phytophthora ramorum, and the pathogen produces abundant sporangia on the needles, which can then spread in wind and rain. And this top right-hand photograph shows the sporangia being formed on the needles. The pathogen can spread very rapidly in epidemic years, and so far over 11,000 hectares of larch have been killed or felled in the UK due to Phytophthora ramorum. Now, my PhD student Heather Dunn looked at the epidemiology in the field of this pathogen on larch over a few years and found that it starts off by infecting the fine shoots through the needles and then causes lesions which extend down the branch systems and causing these girdling cankers on the main stem. And you can see this lower left-hand photograph shows the bark being cut away to show the lesion underneath and lots of resin bleeding. And then the lower right photograph shows the typical start of the infection as it comes in at the top of the tree and spreads down. So the loss of larch is having a very significant impact on the UK production forestry. In the UK, we really only have 2 viable timber species on our upland acid sites on the west of the country, which is basically the predominant areas of forest production in the UK. The other species is Sitka spruce. So if anything were to happen to Sitka spruce now, we would really be struggling. So the forestry sector has a great interest in retaining larch as a viable timber species. And so they were able to give us a very small amount of money to start looking into whether there was any natural resistance in Japanese larch. And one of the reasons we were able to do this, because in the rest of the country, as soon as larch is infected, it's felled immediately for statutory control purposes. But in the spring of 2013, the Phytophthora ramorum epidemic on larch in southwest Scotland became visible. It spread into southwest Scotland at this time. And the top photograph here shows, it's quite a famous photograph now. It's been, you see it on a lot of presentations, and it was taken obviously from a helicopter. And you can see the browning here is the Japanese larch, which is not flushed in the spring because it's dead. The brighter green trees are the larch, which is flushing, and the dark green is Sitka spruce. But you can see this, the previous year these trees were all healthy, and then this year they're dead. So there was so much larch infected at that time in the management zone that they just couldn't implement immediate statutory felling. So they created what was known as a management zone, which allowed for the phased removal of infected larch stands, and the retention of these stands allowed us to carry out this research. So as I said, the first wave of the epidemic came through southwest Scotland in the spring of 2013, and 2 years later, in stands that were still standing, where you have more than 95% mortality, you have also, we saw that you get these individual trees which are still surviving infection. And so we wanted to ask the question, are these just disease escapes, or are they naturally resistant? So in 2015 and then early 2016, we collected scions from 10 survivor Japanese larch trees and 2 stands in the Galloway forest. And we also collected scions from 14 infected trees in a nearby stand. So infected trees being trees where the tops had died back, but the lower crown was still live, and we were able to collect shoots. The provenance and genetic variation within the sampled Japanese larch stands is unknown. These were grafted by Dave Clark, our nursery manager, at NRS onto European larch rootstock, and they were grown on in a polytunnel here, which was put up in the management zone for this purpose, because we weren't allowed to bring the material back to our labs, which is outside of the management zone. So then Heather Dunn, PhD student, inoculated excised shoots of these grafts 4 years later when they were well grown in October 2019, and the entire experiment was repeated in August 2020. She had to inoculate excised shoots and not whole plants because, again, the quarantine regulations were such that we weren't allowed to bring the material over to our labs if they were whole plants, but we were allowed to bring excised shoots if they were triple bagged. So we have a quarantine greenhouse at our research station that we could do the inoculations. So she inoculated 20 clones, larch clones. So a clone being an individual parent tree from which the grafts were taken, and these included 10 of the known susceptible clones where the tops had died back, and there were also 10 putatively resistant clones. So those were the clones that were sort of survivor trees surrounded by dead trees in stands. She inoculated 6 to 8 replicate shoots per clone with an EU2, which is a local lineage isolate which she had collected the previous year from the area. And she inoculated with a sporangial suspension onto a wound. This was 3 replicate shoots per clone. Sorry, this was 6 to 8 replicate shoots per clone were inoculated, and 3 replicate shoots per clone were just inoculated with water as a control. And these were bagged and incubated at 15 degrees for 21 days. Quickly running through the results, she found that lesions developed in all shoots that had been inoculated with Phytophthora ramorum, and the lesions were ranged from 2 to 240 millimeters in length. And you can see this lower photograph shows a typical extending chocolate brown lesion in the phloem, which is a sort of similar color to the outer bark, but you can see here it extends from here to here. In the controls, there were just some small, what we would call non-typical lesions, just some browning really associated with the wound. So when she did an analysis of the lesion lengths and took the 2 different groups being resistant, putatively resistant, and those which we know to be infected, if you looked at the groupings, they weren't significantly different in terms of lesion lengths, but were close to being significant, and it was a p-value of 0.07. But when you look at pairwise comparisons among clones, there were differences. So this graph shows mean lesion length on the y-axis and the different clones listed across the bottom. Anything that begins with S, SUS was known susceptible to have been infected, and the ones with RES are the ones we think might have some resistance? Well, you can see that there is certainly a gradation in lesion lengths as we move along, and this was consistent across both trials. So obviously this is very early stages, but the continuum of lesion lengths does suggest that there is some variation in resistance. Now, there was another massive expansion of infection in spring 2018, and we were able to go out and look at more stands in 2020 and collected we collected material from 43 more survivor trees, and Heather inoculated these earlier this year with similar results. So clearly there's a lot more work to do. What we have done is this previous set of grafts were planted out in a field trial at an infected site, a site where the larch had been felled due to Remorum infection, and we planted the grafts out there to assess their performance under conditions of natural infection. And actually, I've just been out at that site in fact yesterday, and this is a photograph of the trees. We've relabeled them, so the grafts have actually grown well. But looking at this lower photograph, Phytophthora remorum is starting now to come in to the trial. So we will be able to go out next year and the year after to look to see how that resistance might hold up under natural infection. Now, we have some good buy-in from the local forest industry, and that they're providing us with another field trial site to plant out all the grafts collected so far. So it'll be about 750 trees in a clonal trial, which potentially could be also used as seed orchard if further work looking at whole plants and different isolates of Remorum does indicate that there are differences in resistance. Now, clearly our inoculation method where we wounded is overloading the tree also in favour of the pathogen. So we do need to look at infection through intact bark or through buds, which is where what we believe is happening naturally. And this short paper on this has just been published in Forest Pathology. Okay, so I'll now move on to talk about the second host pathogen system. This is Phytophthora ostrasedri on juniper. It's another emerging invasive pathogen which we believe might have been introduced to the UK in the 1990s. It's soil-borne and primarily infects the roots. Unfortunately, it is now widespread in juniper populations in northern Britain, and it's a real example of an impact of an invasive Phytophthora on a native tree species. Now, juniper is not a commercial species, but it's one of only 3 native conifer species in the UK, and it is of very high biodiversity value. And these photographs are of the same site. It's a site of special scientific interest, juniper woodland in Perthshire in Scotland. The top photograph was taken in the 1970s before Phytophthora ostrasedri came in, and the lower photograph was taken about 4 years ago from a similar perspective, just showing you the devastation that this pathogen is causing. Anyway, we can see that this Phytophthora ostrasedri causes high levels of mortality in juniper, but again, some individuals do remain healthy. So we ask the same question: are these disease escapes or are they naturally resistant? So in 2015, I collected cuttings from survivor trees at 2 highly infected juniper populations. This is a typical example of a survivor tree. One was in Scotland and the other population was in England. And then our nursery manager, Dave Clark, was able to root the cuttings, and they were grown on for 4 years, again in a clonal trial. And this trial was inoculated in 2019 and repeated in 2020. And this time whole plants were inoculated in a quarantine greenhouse with mycelial plugs of Phytophthora ostrasedri. And we inoculated 17 juniper clones from the field, as well as 1 known susceptible juniper clone, which we call JC for various reasons. We inoculated 4 plants per clone, and also 1 plant per clone was non-inoculated as a control. And as I said, the experiment was repeated twice. The experiment was carried out twice. This just demonstrates, these 2 lower photographs demonstrate the inoculation method with removal of a little plug of bark from the lower stem, placement of the mycelial plug in, and then wrapping it up. It's a very consistent, again, it's not the natural system, but it's very consistent, it gives very consistent results. And then we measured the length of lesions after 6 weeks, and it just to show you some of the variation. This was a control where you've just got some staining around the wound, and these two clones were clearly quite resistant to the pathogen because you've got very little or no lesion development. Then you have a clone that's moderately resistant. Just to see the lesions ranging about 25 millimeters. And then we have our very susceptible clone where the lesion has run to over sort of 130 millimeters here, which is similar to our known positive control, which is another clone that we know is highly susceptible to Phytophthora ostracedrae. And again, looking at the results on the graph with mean lesion length on the left and the name of the clonal on the bottom, you can see this variation in lesion length, where some clones were not significantly different from our positive control, and other clones were clearly quite resistant to the pathogen. Some of the clones where we had enough plants, we were able to inoculate them with a second isolate, and this provided some interest because this clone here was actually highly susceptible to one of the isolates and quite highly resistant to the other. So that was a very unexpected result, and we are doing further work on that. So conclusions are that the juniper clones exhibited varying degrees of susceptibility, with some being resistant. So we have a PhD student, Daisy Crowson, who started a year ago, and she is working on a provenance progeny trial, which we also built up from 2015, where we collected seed from 15 different juniper populations across the UK, from 10 different parent trees per population. And the yellow stars on this map show you the location of those populations. So those trees have now grown on, those seeds have now grown on. This is the trial being laid out. The trees were just being removed from the polytunnel this year. This is just a small section of the trial, but it's a great resource for Daisy to use. She's going to carry out a dual RNA-seq trial this year, looking at genetic responses in our known resistant and susceptible clones. But we also want to know if the resistance that we have discovered in the population will allow population recovery. Is it heritable? So that's why we need to look at the provenance progeny trial. So by inoculating offspring from different individuals, we will be able to determine whether that resistance is indeed heritable and can be used in population recovery. So in terms of management considerations, Phytophthora ostracedrae, we know from a lot of work we do on nurseries that it spreads in the nursery trade in a range of different species, not just juniper. And there's been quite a lot of, historically, a lot of supplementary planting of juniper populations in the UK has gone on over the last 30 years. And there is a link between planting at sites and disease outbreaks. So we believe that's how the pathogen has been introduced into the wider environment. And we know that the introduction of more pathogen genotypes through planting could put juniper populations at greater risk. So we are now working with site managers through a juniper group that we have, that's UK-wide, where we're asking them to avoid planting existing viable juniper populations and instead manage them for natural regeneration. And this top photograph is showing one of our most heavily infected juniper sites, but on the drier parts of the site, you have got this natural regeneration coming in, which appears to be growing well. And I just want to, this is a paper that was published on the juniper work a couple of years ago. And one thing we also found, just quickly, I'm sort of at the end of the talk, but one thing we also found is juniper management guidelines in light of Phytophthora ostracedrae have been, we've written these in 2013 and again in 2017 and put them on the internet. But another PhD student, Flora Donnell, did a survey of stakeholders and found out that very few of them actually read the guidance or followed the guidance or even knew of its existence. And so what we've done, and those who did read it found that it was too vague, too complicated, far too long, and didn't really suit their needs. So what we're doing now is working with the stakeholders to co-develop guidance that they will find useful and that they have buy-in with. So at the moment we've produced a two-sided flyer with some of the key considerations, and we will be updating the previous juniper guidance as well. So it's really about working with the stakeholders to find out what, why they do what they do and how they could do it better. And with that, I would like to thank you very much.
Thank you very much, Sarah. I hope you can hear me.
Yes, I can. I hope that was all right, the recorded talk. Sometimes recorded talks get a bit tedious. I kept it short. I kept it a bit shorter so I thought there might be a bit more time for kind of interaction and questions at the end. But,
well, it was very...
I liked it.
I liked it. It was very interesting, and there's lots of relevant learnings for the situation in Australia. So I believe how this is going to work is that the audience will ask questions. We're not sure if you can hear the audience, and then I might have to relay the questions to you. So any questions from the audience? Tom?
Are there any
Sarah, were you able to hear that?
No, if you could repeat that.
Okay. Yeah, no, that's fine. Just checking. Question from the lovely Tom. Were there any traits of resistance in the resistant varieties that stood out to you?
No, not that we could see. Juniper is, you mean morphological traits, obviously. Yes. Yeah, no, and juniper is extremely variable in its growth form. It's very high genetic diversity and very variable. So no, we weren't able to do that. The only trait that we've checked for so far is male versus female trees, which didn't appear to have any difference. But again, this is still quite early stages. But we're hoping to be able to build a genetic understanding of the basis for resistance. But in terms of selecting trees, I think because that's what people ask, can we select trees and breed from those trees for resistance? And I think we could, even though it's early stages, we could fairly, with some degree of confidence, say that within any natural juniper population, you will have natural resistance at some degree, and so it's a matter of actual managing the site for natural regeneration rather than planting.
Excellent. Giles, you had a question?
Thanks for the talk, Sarah. The very hot weather you've been having in the UK, has that exacerbated deaths? You see more deaths as a result of heat waves and the drought effects making symptoms worse?
Yeah, I did hear that question. Thanks, Giles. And well, to be honest, we didn't have the severe temperatures in the north of Britain, and Phytophthora ostracidri is a cold temperature pathogen. So I can't say at the moment that we've seen any evidence of increased deaths of juniper. Most of the infected populations are in the north of the country rather than in the south, and that's where Phytophthora ostracidri is most prevalent. But actually, I was down in the south of England for a few weeks doing field work earlier, and the effect of drought on other tree species was very, very apparent. So that's going to be quite interesting going forward, whether there's been any long-term effects on other tree species.
Sarah, thank you very much. We'll better leave it at that for we're running a little behind schedule, but a big round of applause for Sarah.
Thank you. Okay, well, thanks very much. I've now got to go into NRS. We're having a demonstration of digital PCR. Yeah. So I wish you all the best for your event today, and thank you again for inviting me and for the very, very lovely introduction, Peter.
All the best. Thank you, Sarah.
Okay, thank you. Bye-bye. Bye-bye.
I'd encourage you all to stand up. It's been a long day, and if you want to stretch it out, it's a good thing to stretch. I have a